Hello, my name is Ty Blount, and today I'll be talking to you about RNA synthesis in E. coli. We'll start off with a brief overview of what transcription is, mostly review what RNA polymerase is, and then conclude with transcription in E. coli. Let's get into it. Alright, so transcription is the conversion of DNA information into its RNA complement. And just as an aside, a good way to remember the difference between transcription and translation is, let's say that you miss class once or twice, and so you're copying someone's notes down. That would be like transcription. Whereas if you were copying someone's notes down into a different language, say Russian, which uses a different alphabet, that's translation. You're translating from one language to another, whereas in transcription, you're just translating between languages, if that makes sense. So DNA gets transcribed into RNA between the same language, and then the RNA gets translated into an amino acid sequence, just between two different languages. Hopefully that helps a little bit. Anyway, back to what we were talking about. Messenger RNA holds the code for an amino acid sequence of one or more polypeptides as specified by a gene, and then transfer RNA gets read by mRNA and then translates the nucleotide codon to its complementary amino acid, transfers it to a growing polypeptide chain after that. Hopefully this is all review. We'll jump into something else. Moving into DNA-dependent RNA polymerase, let's talk about some of its requirements. It requires a DNA template. All four of the ribonucleoside 5' triphosphates, or NTPs. We know them as ATP, GTP, UTP, and CTP. And then also magnesium. So let's break that down. Alright, so as we just mentioned, RNA polymerase requires a DNA template, which can be seen right here. It requires nucleoside triphosphates, which one of the examples could be right here. And it requires magnesium. So let's talk about magnesium first. Magnesium ions will be found bound to aspartate residues within the RNA polymerase. So magnesiums are bound to aspartates from the polymerase, and what they do is they help stabilize this triphosphate right here. Alright, and so what happens is these two electrons attack this phosphate, and then this bond right here moves over onto this oxygen. And so we have an inorganic phosphate leaving the system, and this new nucleotide being added to the RNA. Real quick, back on these magnesium ions. One magnesium ion facilitates the attack by the 3' hydroxyl group on this alpha phosphate. So that would be this magnesium ion facilitating this attack right here. And then the second magnesium ion facilitates the displacement of this pyrophosphate. And both metal ions stabilize this entire transition state. So real quick overview, what's happening. Here we have DNA, and RNA polymerase is adding on these nucleotide triphosphates to a growing strand of RNA that's growing from 5' to 3'. And each time one of these triphosphates is added, it's facilitated by these magnesiums. And what happens is the lone pair of electrons on this oxygen attack the alpha phosphate, and then this inorganic phosphate will leave Okay, so a quick overview of what just happened. RNA polymerase builds the RNA 5' to 3', which means that the DNA gets read 3' to 5' naturally. The 3' hydroxyl attacks the alpha phosphate of the incoming NTPs in order to add it to the growing RNA. And it could all be summed up in the equation NMPN, which is our growing RNA chain, plus an NTP, plus a triphosphate, gets converted into NMPN plus 1, which is basically we just add the nucleotide onto our RNA, and it grows, and an inorganic phosphate. So let's take a look at this a little bit different way, real quickly. All right, so here we have RNA polymerase. It's kind of like a side cut of it, vertical cut, I don't know how to put it. But what we have is going to be some DNA, right? And we'll have it enter and kind of split apart. And so right here, in order for transcription to occur, this DNA has to open up naturally. And this is what's called a transcription bubble. Down here at the bottom, we're going to have the RNA exiting, going this way, and being formed over here. So over here we have our nucleotide triphosphates coming in, NTPs, entering the RNA polymerase, and forming right along here, kind of like this, let's say. And this is our RNA that's leaving the RNA polymerase. So it's forming right around here. And as you can see, a little bit, you can't really see with my drawing here, but during elongation, RNA will temporarily form a double helix with the template DNA strand, which you can sort of see right here. I'll draw little lines like this to make it more obvious. And so shortly after its formation, though, this hybrid duplex, let's call it, will peel off, and the RNA will go on its own way. And the DNA will reform its own duplex. And so because of the transcription bubble, the DNA will need to get rotated to open up. So it's kind of like if you have something like this, it's going to need to open up and unrotate it, if that makes sense. And so then what happens is we have some positive supercoils that form over here on the exit side of the RNA polymerase, and so tightly wound, and then negative supercoils that occur over on the entry side. And so these are looser, sort of quote-unquote coils. That's way too many lines. Let's just say that makes sense right now. So we have positive supercoils forming as the DNA leaves the RNA polymerase, and negative supercoils forming on the side that's entering the RNA polymerase. And just as a quick review, these supercoils, they don't help the DNA out too much, so a topoisomerase will come in and snip up the phosphate backbone, allowing it to re-rotate back to its natural sort of state, if you will. Let's go over some numbers for the E. coli RNA polymerase. So this bubble that gets formed in the E. coli RNA polymerase is about 17 nucleotides large. So there's like 1, 2, 16, 17 nucleotides that detach in order to form this bubble. And then within this bubble, in this area right here, we have 8 base pairs that form between the RNA and the DNA. And so this is the hybrid duplex that in E. coli will form 8 base pairs between the two as this RNA is forming. And this elongation process of RNA occurs at a rate of around 50 to 90 nucleotides per second, according to the book. And just real quick, we'll go over the two roles of the, or the role of the different strands of DNA. We have the template strand right here, and the non-template strand. Obviously, template strand is the one that all the NTPs will come and bind to. And then the non-template strand will have a nucleotide sequence identical to the forming RNAs, except for you just have to swap out the thymines with the uracils, because that's how it works. Alright, so let's talk about RNA polymerase. The holoenzyme is composed of 6 main subunits, 2 alpha subunits, an omega subunit, a beta prime, and a sigma, which all probably means nothing to you, so let's draw it out real quick. First off, we'll start with one of the beta subunits, which will kind of be like this. I don't know. Lima bean? Is that what lima beans look like? Maybe that's what lima beans look like. I can go with that. So this right here, for now, is going to be our beta subunit. Attached to the beta subunit is going to be our beta prime subunit. We'll draw this kind of like this. There's an opening over here on the right. Might be your left. Alright, but over here we have a opening of sorts. And it's kind of like an oven mitt, maybe. Yeah, it's like an oven mitt. And this other one right here, you can think of them kind of being like a claw, right? And so we're going to have everything feeding through this entry in the claw. The other subunits mentioned before were 2 alpha subunits, which we'll draw kind of like as little p's over here. Alpha, alpha. And then an omega subunit, which we'll draw, I don't know, just attached down here. Omega. Right? So this is the main makeup of the RNA polymerase. Now I mentioned that there were 6 subunits, and the 6th one is a sigma subunit, and it's kind of the one that is, I don't know, seems to be the most important one. It isn't directly attached to this at all times. It acts more like a cofactor. But when it is attached, it will attach right here. And that is a sigma subunit. So real quick overview. This is our RNA polymerase. We have our alpha subunits, an omega subunit, a beta, and a beta prime that form this kind of claw-like structure. Our RNA is going to be synthesized, and the DNA is going to be entering all in this little pocket right here. And then we have a sigma subunit over here on my right. And this, as we'll talk about in a little bit, helps guide this RNA polymerase to the portion of the DNA that it's supposed to get to. Alright, so now that we've talked about RNA polymerase a little bit and its structure, let's get into actual transcription.
This is a really simple sort of diagram I drew up. Honestly, should have been an art major probably. But we have RNA polymerase, which comes in and, guided by its sigma cofactor, will nonspecifically bind to our strand of DNA, which I hope is obviously this double-stranded helix. RNA polymerase comes, nonspecifically binds to the DNA, and this sigma cofactor will help it identify the proper promoter. Alright, so after it's moving along and does identify its promoter, or the promoter that is available for it, what happens is we have our transcription bubble form. So you can remember again, there was kind of this little middle subunit right here, and this RNA polymerase, just as a reminder, is bound with its claw, the two beta subunits facing down in this position, or from this perspective. So we have a transcription bubble form after the promoter has been found. So it begins to unwind, and it moves from a closed complex to what's called an open complex. And transcription is initiated within the complex, and it converts it to its elongation form as it moves away from this promoter. And as it enters elongation, the sigma subunit will fall off. Now it is competitively beat out by a protein called NUS-A. It stays in contact throughout elongation. So it's just another cofactor that's bound to the RNA polymerase throughout the elongation step. And so after elongation is over, it'll dissociate and then gets replaced by another sigma cofactor, whichever one happens to be more present within the cell at the time. So next, let's talk about sigma subunits a little bit. In E. coli, there are 7 different sigma subunits that are used. Here we have 5. The other 2 occur in very low concentrations or ratios of molecules per cell. So on the left here, we have the different subunits. In the middle, we have the amount of molecules per cell out of 1,000. So for example, the sigma 70 has about 70%. That's not why it's sigma 70, but you get the point. And then on the right here, we have the different functions of these sigma subunits and what they're used for and what they're used to guide the RNA polymerase to. Alright, so let's talk about the functions briefly. Sigma 70 is most prevalent in E. coli, and it functions to take RNA polymerase to activate genes that, as the book puts it, are in charge of the housekeeping of the cell. Sigma 54 is in charge of the nitrogen levels, keeping those at proper levels, keeping the nitrogen at proper levels, excuse me. Sigma 32 is in charge of the heat shock genes. So if a cell experiences a sudden increase in temperature, then sigma 32 will be able to bind to RNA polymerase and take it to the correct promoters to increase different things that the cell can use to not combat the heat, but to deal with the heat. Sigma 28 is used for flagella and chemotaxy, so different movement options of these bacterium. And sigma 24 is extracytoplasmic functions. And it also includes, or extracytoplasmic functions also included in the other 2 sigma subunits that aren't mentioned here. Right. So why use different sigma subunits? I think this gives you a pretty good picture that each subunit will select for a different function, or vice versa. Each function has a subunit that will select for a promoter that corresponds to it. By using different sigma subunits, a cell can coordinate the expression of sets of genes as well, permitting major changes in cell physiology. So the sets of genes that are expressed is determined by the availability of various sigma subunits, which is determined by several factors: regulated rates of synthesis and degradation, post-synthetic modifications that switch individual sigma subunits between active and inactive forms, a specialized class of anti-sigma proteins, each type binding to and sequestering a particular sigma subunit. And so the sets of genes that do get expressed, those are determined by basically the availability of these different subunits. And all of this is regulated by several different factors. Either the rates of synthesis and degradation are regulated, post-synthetic modifications that switch individual sigma subunits might be active or to the active and inactive forms, and a specialized class of anti-sigma proteins, each type binding to and taking that sigma subunit out of the selection pool to be used for the RNA polymerases. So next we're going to move into the different forms of regulation. So let's briefly talk about activation and repression in E. coli. The binding of proteins to sequences around the promoter will affect the rate of transcription of mRNA. In E. coli, one protein that activates transcription is the cyclic AMP receptor protein, which increases the transcription of genes coding for enzymes that metabolize sugars other than glucose when cells are grown in the absence of glucose. So when glucose levels are low, this receptor protein is used to activate the transcription of genes that code for enzymes that will work with sugars aside from glucose. Repressors are proteins that block the synthesis of RNA at specific genes. And so in the case of the lac repressor, transcription of genes for the enzymes of lactose metabolism is blocked when lactose is unavailable. Which makes sense. No lactose metabolism. Moving on. So let's talk about termination and termination signals. RNA synthesis is processive, so RNA polymerase will introduce a large number of nucleotides into a growing RNA molecule before dissociating. Termination doesn't happen at a specific instance, if that makes sense. So it won't just get snipped off at a certain sequence. And this is all necessary because RNA polymerase can't rebind to DNA. Let's say the polymerase stops prematurely. It won't be able to fix the mRNA. And so there's kind of this gap that's added as a safety. Better safe than sorry. And so next we're going to focus on the two ways that termination occurs in RNA polymerase in E. coli specifically. So these two classes of termination signals are called row dependent and, might have guessed it, row independent. That's not how you spell it. Oh, I'm messing up a lot. Independent. Dent. Alright, let's get into it. So first let's talk about row independent termination. There's a couple distinguished features. It produces an RNA transcript with a self-complementary sequence, and it leads to the formation of a hairpin structure. That's the first feature. So we have our U's right here, the uracil, that will end up complementing itself and creating this hairpin-like feature. And this is because of a highly conserved string of adenine residues in the template strand. So it gets transcribed into uracil, and when the polymerase reaches this section, it results in a isomerization of the RNA and the polymerase, actually. The hybrid region at the 3' end, the transcript is relatively unstable, and the RNA will dissociate from the complex completely, leading to termination. Now let's talk about row-dependent termination. Unlike row-independent termination, a hairpin-like structure isn't used to dissociate the mRNA from the RNA polymerase. Instead, something called a RUT site, which stands for row utilization. Row utilization element will bind, it will bind to row helicase, and the row helicase will come in. Call this row helicase. It will migrate along the mRNA to the elongating RNA polymerase. And when it reaches it, it'll separate the mRNA from the RNA polymerase. So just to add in this row helicase right here, this ADP is ATP dependent and promotes translocation of the protein along the RNA.